followed by a presentation by our scientists. Then after the presentation, Dr. Zwelini Mkhize, the Minister of Health, will address us as well. Then we'll take question and answers from members of the media. For the question and answer session, we advise that members of the media send their questions to the Q&A session. We will attempt to respond to all of those questions. For those questions that might not be fully or comprehensively addressed today, we will be sending responses to the various media houses. With those few words, I would like to welcome you all, and I would recognize Minister Nzimande to open this briefing for us. Minister Nzimande, over to you.
Thank you. Thank you very much, Ishmael. I hope everyone can hear me. Let me start by greeting my colleague, Minister Zwelini Mkhize, the Minister of Health, and also greet Professor Tulio de Oliveira, who is the leader of this team, together with the other scientists and researchers who have joined us, members of the media, ladies and gentlemen. I have convened this briefing together with Minister Mkhize today so as to share with you some of the new scientific findings from the work that has been funded by our Department of Science and Innovation in support of the fight against COVID-19. To show, as we were going to do today, in the main, that my department is fully part of this fight against COVID-19. You may also recall that if last week I hosted a virtual COVID-19 conference whose aim was to inform the public better about COVID-19 and the role of science, including vaccines, in the fight against this pandemic. Government has, through the Department of Science and Innovation, been working around the clock together with our scientists in the fight against the pandemic. And I would just like to highlight, by way of background, just a few things that we've been doing so that I contextualize this work properly. Firstly, our country is fairly well respected globally for its relatively advanced capacity in the field of science, technology, and innovation. In fact, in some fields, we are very advanced. Our scientists and researchers have rolled up their sleeves and collaborated even with their global counterparts to find solutions to save humanity against many diseases, not just COVID-19, but obviously including COVID-19 at the moment. In this regard, our researchers and facilities have contributed to the detection of new variants of the novel coronavirus. When I convened last week the COVID-19 webinar and conference, I also indicated the extent to which the Department of Science and Innovation is also funding research into genomics, the study of genetic types with a view to support our fight against COVID-19. And to this end, we've spent 69.4 million rands in funding for COVID-19 research and innovation, covering a total of 21 projects. I had indicated last year that when coronavirus variant 501Y-V2 was discovered by our scientists based at the KwaZulu-Natal Research Innovation and Sequencing Platform, this work was supported and funded by our Department of Science and Innovation. Because of that research, we now know that individuals who happen to be infected with the 501Y-V2 during now the second wave of COVID-19, which is what we want to share with you today, would have developed antibodies against reinfection, or possibly by pre-existing variants. We will actually be giving you details on this score. But I also want to use this opportunity to point out that there is no doubt in my mind that with the work that our scientists are doing, we are heading in the right direction insofar as deepening our knowledge and getting closer to dealing with the enemy before us. That is why, therefore, today we have convened this to announce that we've notched another important scientific discovery, which in short is that the research done by the KwaZulu-Natal Research Innovation and Sequencing Platform, KRISP, working together with the National Health Laboratory Services, African Health Research Institute, and CAPRISA, as well as the National Institute for Communicable Diseases, have come out with the findings which have demonstrated that those who were infected in the second wave, which was now dominated by what we have been referring to as the new variant, 501YV2, have protection from current and previous circulating variants. The 501YV2 variant is able to generate immune responses, we have now found out through our scientists, that neutralize itself and other SARS-CoV-2 lineages. What this means, amongst other things, is that vaccines now that are based on dealing or likely to generate neutralizing agents against the current variant can actually be developed given these findings. I am nevertheless not going to be the one to give the details about this, but this is a highly significant finding by our scientists in the fight against COVID-19. Let me then, with this brief background, hand over to Professor Tulio de Oliveira to introduce his team and then to make the presentation as indicated by the program director. Thank you very much.
Good day or almost good afternoon. First to our Minister Nzimande, thank you very much for this very kind introduction. Also to our Minister of Health, Zwelini Mkhize, and DGs presented. So today, what we come here to you is just to keep providing very clear information on how the science is advancing very fast in South Africa. One thing that we did in the beginning of the pandemic, and is going to be a year that touch our shores next week, is that we decided that a large number of organizations, both in many regions of the country, decided to pivot. What it means, we decided to work together and put our resources to try to understand this virus that was touching our shores, which we knew that had a great pandemic potential, as quick as possible, with the objective to really follow that and try to use the scientific knowledge that we have to understand, but potentially to try to control the pandemic or to develop better interventions. So that's what I will tell you. Today, what we're going to be sharing, it is potentially a relatively good news or scientifically very good news, but we're going to put that in the frame of the public. For us, it's very important because sometimes we felt that our main job as a scientist, or especially at the Network for Genomic Surveillance, a lot of time was to give bad news to South Africa. So it's our great pleasure that we keep persevering and working very hard to try to understand this virus in real time. Yeah. So one thing that is very clear is that this advance in science doesn't happen alone. So what we did is to work with a large number of organizations. And today we have the great honor to have 3 other professors with me, and we're going to do this presentation together. We're just going to pass one to the other, that we're going to comment in each other's work, but should be a continuous process. So today I have Professor Zwelini Mkhize join me, Alex Siegel, and Penny Moore. And just to highlight is the number of different organizations that are working together to try to face that challenge in South Africa. So this presentation is going to be short, should be around 15 to 20 minutes max. And what we're going to touch in 3 aspects that we think that would be interest, not only for the scientific community, but also for the public in general. Yeah. So we're going to give a quick update of the network of genomic surveillance. That's what NGSSA means. And the variants in South Africa, we have found more than one, but that's going to become quite clear. And then we're going to highlight some of our work that was crucial to understand how the 501YV2 effect of neutralizing of previous virus and vaccines. That was crucial to do that quite well and quick, especially to guide the rollout of vaccine in the country. And then we're going to give you the new information that's about the effect of the 501V2 to also producing high-level titer antibodies that could neutralize itself very strong. We expect that, but in this virus, it is a lot of things is unexpected. So it was great to confirm that. But the new news is that it can also neutralize better than the previous variants, the other variants, and we're going to go there. So I would like to please ask to Professor Zwelini Mkhize just to explain the next 2 slides, because it was really, really important how we did that, not as a just group of academics, but also together with our national laboratories.
Thank you, Tulio. And good morning, good day to Minister Nzimande and Minister Mkhize, and to all the panelists as well as those that are attending this webinar. Just very briefly, as Tulio has outlined, so what actually happened, especially within KZN, this was around the time where we had the hospital outbreaks, and everybody will remember that we had, an outbreak with, in one of the hospitals in KZN around April of last year, where then the samples from the NHLS laboratory, the Viral Academic Lab in KZN, submitted samples to Tulio's group where the genomic analysis was done. This was then subsequently followed with a systematic submission of samples. So what happens within KZN, for example, is that every week the lab, the virology NHLS lab submits 50 samples, which actually are randomly selected samples representing all the districts in KZN. These are submitted weekly to the lab, and the analysis is done within 7 days of sample collection. And this then got extended to other national health laboratory labs across the country. As you can see in this slide, in that Bloemfontein, which is Universitas and University of Free State, as well as the 2 hospitals in the Western Cape, Tygerberg, as well as Grooterschuur, and the NICD in Johannesburg. So we have this complex and network of labs that submit samples, which then get analyzed over the time period. And this has been happening since basically May of last year. And if we just move to the next slide, Tulio. So what this has actually allowed is that we try to make sure that as samples come in, the sequencing is done as close to sample collection as possible. As I've already mentioned, for instance, in KZN, this is done within 7 days of collection. And you find that in the various areas where this is happening, we try to keep it as close to the, to sample collection as possible. And, whilst I'm just talking about NHLS samples, at the the moment, we actually have got about 5 to 10% of samples that are coming from the private labs. So this gives us a good overview, because each and every lab that submits samples, they actually make sure these samples are representative of the districts within those provinces. This as well is obviously assisting in that we have managed to define and we've managed to support the relevant public health priorities, in that we are now able to sort of define and describe any clusters of outbreaks. And we have seen this working very well in evaluating vaccine rollouts to understand which vaccines will work in the face of the variants that are emerging in the country. And it also has created a good platform where bioinformatics actually we can get a better understanding, which is not just from one province, but it actually gives us a national analysis of the data within the country. So we've managed as a result to produce these, timely reports, which have assisted, the ministers to make decisions, and we therefore are able to actually inform the responses to the pandemic as close to real time as is possible. Thank you. Let me hand over to the next speaker. Thanks, Tulio.
So thank you, Koleka. Yeah, we thought that was very important to both our media but our public to know of this very close relationship between academic labs, 5 genomics facility producing data in real time, and how we use that to inform. So you may have seen this graphic. We keep updating every week. Yeah. So here, what it means, we have it on the x-axis, we have the dates. So we start in February, March last year. We even sequenced the first contact in the country, and we do that every week in the country. In the y-axis, we have the percentage of the genomes. When we have white, what it means doesn't mean that we have data, normally mean that we have 20, 30, 40 different lineages of variants circulating. And then what we see is that around the first wave, the lockdown, we start getting our own specific South African variants. They got numbers in the classification, but they never took over completely until the appearance of the 501YV2, which tended to basically extinguish or decrease it to very low percentage the other variants and start dominating the pandemic. So we could do that, and we continue doing that every week. Yeah, these results, it's used to inform not only our health department, but also we work closely with the Africa CDC to support the neighboring countries, so we can know how fast and how far this variant is spreading. Yeah. If you go to South Africa, this is new data that we're presenting just now. What you see is this very starking growth of the proportion of the genomes that are of this variant. So the 501YV2 also go by a second name. That's a different classification, but it's the same variant called B1351. And then what we see that since October, the proportion of the genomes increasing from 10% to almost every new genome that we produce is this variant. One thing that allows us to do also, because now, given that we have a high proportion of the 501YV2 dominating our pandemic, we can get very close eyes and very, very focused on identifying the different sequence. So what we have done, and we have reported that around the end of December, beginning of January, we found the first introduction of this other variant called the B.1.1.7, and that's the one that was discovered in the UK, and that's causing much fast transmission. However, it is only one introduction, and so far doesn't seem to be spread at the population. Last week, we also reported that we found another variant that's called the A23.1. Yeah. This is a new variant, is not yet of concern what it means. We are not so sure how dangerous it is, but this is originated in Uganda and it's also transmitted a lot in Europe. So both of them is just one sequence, and they will fall either in January, that's why you have that distinction. But what we found is that our variant is still dominating very high. But this is what the genomics surveillance allow us to be picking up new variants all the time, and we're going to keep a look if they grow in prevalence. Yeah. One thing that we know is that the 501YV2, the one that was firstly identified in South Africa, which is quite clear, we don't even know if it originated in South Africa. We know that was identified in South Africa through this network and spread very fast. Now it has been found in 48 countries in the world, yeah? And again, I know that the media will write, so it doesn't make sense to call the South African variant because we are at close to 50 countries that this variant it is circulating, yeah? One region that we worry is the Southern African region. And you may have seen that beginning of the year, they also had a severe second wave. And some of our neighboring countries, especially the ones on the east, they have very fragile, weak health systems. And they have very large number of deaths. And a lot of those would caused by the same variants that circulate in South Africa. We also found that at a high proportion in Europe and have entered the US and the UK. So what is different about these variants that we are reporting? Because normally we have found for almost a year, 2030, and we didn't report. What's the difference is that they have many, many mutations. So they have many more mutations, and they have mutations where you don't want them to be. More specific, in the most important protein of the virus, the spike protein. So the 501YV2 has around 9 mutations plus a deletions, with these 3 ones being very important, the 417, 484, and 501, because the 501 has been associated with high transmission, and the 484 also with decrease in neutralization of antibody. A new mutation that we are getting worried because it's also popping in the B117 and the P1 in Brazil is the 18F. This one also may increase transmissibility. Yeah. So that's the results. I'm just going to go to the first wave results, and then I'm going to pass to my colleague Alex Siegel to explain that. But what we did in South Africa, especially with Professor Alex Siegel group at ARI, it is to set up these very advanced experiments, just to take simpler to you. So here what you have these pictures, and what's these pictures is the number of viruses that are grown. What we call that's the live virus assay is one of the gold standards. And then what we had like here, what you do, you take the individual infected with a first wave variant, this means not the 501V2, and then as you increase the amount of neutralizing antibody or plasma, you can see you clear the virus, so you can neutralize the virus. This number 150 is very high concentration, 1400 is less. And what Professor Alex Siegel noticed is that in our variant, the 501YV2, as you add more plasma from previous infections, you had some neutralization, but much less. You can still see the difference between the number of virus formed. And that was the results that we give in the beginning of the year, which caused major concern around the world. Yeah. So what we did as a collective group between the Wits University, ARI, and ICD, KRISP, and the NHLS is to work very close to answer some of the most important questions. Yeah, one of them that was, does the plasma collected from people after receipt of the AstraZeneca vaccine neutralize the virus? Yeah, that was very important because that was the vaccine that we were going to roll out in South Africa. And then what we see first on the live virus of Alex Siegel, and he's going to present his new results very soon, is that you had a massive drop of neutralization. 58% of them were not neutralized. And then Penny Moore also confirmed these results with a completely different system, which is very good scientifically when you can confirm results with different system that you call a pseudovirus, that what you show is that big decrease of neutralization. This means not neutralize the new variants with 3/4 of them from the plasma of the vaccine have what we call very low or what called knockout, almost not being neutralized. And this is the work from Shabir Ahmadi, but he approached the whole network to do that very quick. And we could do that as a collective in a few weeks, and that's the power of working as a big group. And you know what's the effect in AstraZeneca? They decided to roll out another vaccine. Just to ground you before we go to the next results, that's a results slide kindly provided by Professor Salim Abdul Karim, is that now in South Africa we have these two systems that we can test the decrease of neutralization of the different vaccines. And what they did, not only us, but across the world, is to show that some of the vaccines decrease neutralization, but not at very high level, and others at more high level. So what it means is that we have now this system, both in the country but internationally, that we can see the effect of the variants on decreasing neutralization of the vaccines. And if you put this together, more powerful, these results with the different trials that are running in the country, you can start doing what Professor Salim Abdul Karim has done very well, is starting to ranking what it is, the potential effectiveness of the vaccines that we can use in the country. And the reason why we start with the Johnson & Johnson single dose is because that one have been trialed in the country, very important to do this process in the country with our population, our variant, and show very high clinical efficacy, especially on hospital and severe. The AstraZeneca not only had a lower fold reduction, but a lower efficacy on the mild moderate, and we are still waiting for the clinical efficacy of the high. So the answer from the system that we have done together in the country is that you could evaluate very quick the vaccines that are being rolled, and to see what's the effect, not only on the lab, but also with clinical trials and roles on the decrease of efficacy of the vaccines, So now I will just pass to Alex Siegel, because this is his very hard work, and I would like him to explain how different was on when we put the virus of the second wave to neutralize the 501v2. Alex Siegel, if you can join.
Hi, Tulio. Thank you very much, and thank you, ministers and the other participants. First of all, I would like to thank a few people. First is Tulio. Tulio had a kind of an incredible instincts with these variants. So he recognized the danger before anyone else and made sure that other people understood it, and really got me on board to investigate this. The other person is Professor Karim, who has been supporting me here for a while now, and helping me to in my career. And third is my PhD student, Sandile Sele. So when these variants hit, it was basically during the holidays. And, we talked about it, and he said, I'm right there, let's do it. And he was the first one in the world to isolate this virus, which wasn't simple. And so we're able to work with it. And he also did all these experiments on which this work is based on. So just a terrific student. He never been out of KwaZulu-Natal, and he made for a starting researcher, he made just an incredible contribution and incredible commitment. Okay, so let me go on kind of to the results. And I have to say, when we got this result, we had to change our personal protective equipment because we're both on the floor. We're just floored, literally floored by this result. So we had the results before that the 501YV2 was escaping neutralization by the plasma from the first wave that was elicited by the earlier variants of SARS-CoV-2. And what we didn't know what's going to happen with this plasma that's actually made in response to this virus, so the 501YV2 that Tulio discovered. And what we found is, first of all, it could neutralize itself. And this is what you're seeing in this set of images. So as you're going from left to right, you're decreasing your dilution, right? So 1 to 25 means you have concentrated plasma. So concentrated, it's still a dilution from what you got in your blood, but not much, right? And as you go now towards the right, you have less and less of your blood in trying to neutralize the virus, right? And what we're seeing with the 501V2 is the predicted effect of good neutralization. So if you have higher concentrations of your blood, therefore antibodies, you'll have no virus because it's being blocked by these antibodies. And as you go to lower concentrations, you get more and more virus. And each one of these dots that you're seeing, it's called a focus. And it basically shows you the effect of one virus. So one virus infects one cell and then infects other cells locally and gives you one dot. We stain it for the virus and we see kind of, okay, we have one virus that actually managed to escape, right? So as you can see, this is the expected effect if you have actually effective neutralization. Now, what was surprising to us is what happened to first wave virus. So we used this plasma diffraction from the blood from people that were infected in this latest wave with the 501YV2. And we used it against the first wave virus, so the earlier variants. And what we found was that it could neutralize. Okay, not as well as it could neutralize itself, right? But it's not bad at all. And of course, this kind of gives you some idea of how this problem of variants can be solved. And maybe it can be solved with the variant that was first found in South Africa. Because it seems like if you actually are infected with this variant, and this, of course, this statement is limited to these results with neutralization antibodies. But if you're infected with this variant, or you can design a vaccine with this variant, it might be cross-protective to other variants. It might leave the virus with not much room to go, right? And this, of course, to us was incredible. So that's basically this piece of data, and this is kind of the summary of it. I won't kind of go deeply into it, but basically what you're seeing here is a bunch of numbers in those colored rectangles. The higher the number, the better. So 344 means it's good. And what that rectangle means is first wave virus being neutralized by first wave plasma, right? So what happens basically in the first wave? So if you were infected with your first wave virus, you produce antibodies to it that were effective at neutralizing it. That's how you got better, or at least one of the ways. Okay, and then the second blue rectangle, the kitty corner there, yeah, that's 501YV2 virus neutralized by second wave plasma. So basically the plasma now in January, so that's also good, right? So these were the infecting viruses and they could be neutralized by people who were infected by them. Okay, that's how they got better. What was interesting is the cross reactions, right? So that's the yellow square rectangles. So the first yellow rectangle on the left, the bottom left, where it says 40, and 40 is not good, is 501YV2 virus against first wave antibodies. So antibodies to the previous variants, and that doesn't work well. But the other rectangle, the other yellow one is much better, as you can see, right? So this is first wave virus neutralized by plasma from people who were infected with the 501YV2. And this works. Okay, it doesn't work quite as well as for its own neutralization for 501V2, but not bad at all. So this is definitely, I think, an important finding and something that I think we and South Africa could contribute to the general understanding of this virus that might be very applicable to vaccines. Thank you.
Okay, thank you, Alexigo. Yeah, very important results. Yeah, and that was done in a research program in KwaZulu-Natal with live virus. So was crucially important that these results could be validated and expanded, yeah, in other areas of the country and with other systems, because normally we trust results that can be replicated with different methods and different cohorts. So just would like to ask Penny more to explain her results, because they completely build on Alexigo's one. Yeah.
Yeah, thank you very much, Tulia and Alex, for the introduction. So as Tulia has mentioned, this is a very reassuring set of data, because what we've done here is in a separate lab using a completely different system and a completely different cohort of infected people have reached the same conclusion, which is something that's very reassuring. So the data that I'm going to show you all in the next couple of slides is another example of a truly wonderful collaboration, a very tight collaboration between us at the National Institute for Communicable Diseases, University of Cape Town, the University of KwaZulu-Natal, and the University of Pretoria. And again, building on existing links that have been built over many years, which enabled us to move really very quickly when this very important question came up. So what I'm showing you here in this slide has already been alluded to. This is the data that we showed you a couple of weeks ago in an earlier press conference. This was data where we essentially took a cohort of 44 people who had been infected with the original variant, and we measured their neutralization, both against the matched variant, the original variant, but also against the new variant. And to our great alarm, and to the alarm of many other people, we saw a substantial loss of neutralization, a 9-fold reduction in average titer, which is essentially a measure of how well neutralizing antibodies can prevent a virus from infecting a cell. So you can see there that this data is summarized in the pie chart at the bottom. And essentially what you can see from that is that using this cohort of 44 people, as many as 48% of them, nearly half of these people, despite having very good neutralizing antibodies against their own virus, were unable to neutralize 501YB2, the new variant. That data was published yesterday in Nature Medicine and was really the beginning of our concern about neutralization escape. So the data I'm going to show you in the next slide has been generated by two very, very talented people, Dr. Thandeka Moyogwete and Mashudu Matsindila at the National Institute for Communicable Diseases. And what they did was to ask, what happens when you ask the opposite question? What happens when you take people who were infected with 501YB2 and you test their antibodies, their blood, against the original variant? Do you see that same dramatic drop-off or not? That's shown in the middle panel, Tulia, if you can use your mouse to show people, thank you. In the middle panel, again, a cohort, in this case, of 55 people. Here, first tested against 501YB2, which is the new variant, and then tested as well against the original variant. And the first thing you can see is that the slope of that red line, which is a sort of a measure of the average response in all of those 55 people, is much less dramatic than the slope in the original graph. You can see it's almost flatter. If you look at the numbers, instead of seeing a 9-fold reduction, which is what we saw in the original cohort, here we're only seeing just over a 3-fold reduction. And what's perhaps more important is if you look at the pie chart at the bottom, only 4% of these individuals were unable to neutralize the new variant, were unable to neutralize the original variant. Even I get confused by this data. So what this means is that, as Alex so clearly explained, is that the neutralizing antibodies elicited by the new variant are somehow different in their ability to be able to recognize not only their own virus, but also other viruses. We started to extend this work. Of course, 501YB2, as Tulia mentioned, is not the only variant of concern. Another variant of major concern is what's called either P1 or 501YB3. And that variant was initially identified in Brazil. And so what we've done here is to ask the same question in a much smaller, unfortunately at this stage, much smaller set of individuals, only 7, but to ask exactly the same question. Are the antibodies that are triggered by 501YB2 able to recognize the variant detected in Brazil? And as you can see in this case, in a small set of 7, they are able to recognize the variant from Brazil. And actually of that small subset, none of them are completely resistant to neutralization. So as has been described, this is potentially good news. it's not that the antibodies that are triggered by 501YB2 are somehow magical. There is a drop-off. They do preferentially recognize the virus that infected those people. But unlike the antibodies that were triggered by the original variant, these antibodies seem somehow to have a little bit more breadth. How they do that is the subject of future work and is important to understand. We need to understand which bits of the spike, which is where neutralizing antibodies bind, which bits are the target of these antibodies, because that will be crucial to understand both in terms of vaccine design, but also in understanding people's risk of reinfection. But the bottom line is that there does seem to be, in people infected with the new variant, a greater capacity to neutralize either variant than what we perhaps expected and what we have seen before. And with that, I'll hand back to you, Tulia. Thank you.
Okay, thank you. So yeah, that's basic the news that we have to give you. All these results are being generated now as we are presenting in the last few days, and we're gonna keep updating, but they are showing a very clear, consistent direction of the variant producing strong antibodies that can neutralize itself and potentially other variants and other lineages of the virus. So just to conclude, okay, so genomic surveillance is a critical component of the epidemic response. Yeah, we have been very fortunate in South Africa to be able to be supported by the Department of Science and Innovation and the Medical Research Council to create this network of labs, and especially fortunate to be able to work very close with the NHLS, so that's done in real time. Yeah. We detected a new variant with multiple spike mutations that it's less neutralized, affected some vaccine response. And for months, yeah, not only scientists, but the whole country was hammered from this detection. Yeah. One thing that we show now, the new results, that the plasma collected from people infected with the variant has good neutralizing activity against itself, but also against the first wave virus and potentially other variants of concerns. Yeah, what it means? It means that people infected may have some immunity against the variant and other lineages, but it also means that we know that immunity can decrease over time. So in no way we are saying that these people shouldn't be vaccinated. The opposite. Everyone, we should try to increase vaccination to avoid that very deadly third wave. Yeah. And that just to highlight that by working all together, yeah, and by getting all these research institutes to work together to answer the critical questions, we really think that at the moment and for the foreseeable future, we have the scientific tools to evaluate new variants and the effects on neutralization and vaccines as they advance, not only in South Africa, but within the continent and the world. Yeah, a lot of time people try to say that this is the work, magical work from KRISP. We would never be able to do that alone. This is the work of hundreds of scientists across many organizations in the country, with strong support from both national and international funders. Thank you very much, and I would like to pass back to our honorable.
Thank you very much, Dr. Tulio. I would like to hand back to Minister Nzimande for his reflection. Then we'll go to the Minister of Health, Dr. Zwelini Mkhize, for his reflection. Then we'll go to the journalist for questions. Thank you very much. Minister Nzimande, over to you.
Yeah, thank you very much, Ishmael, and thanks to our scientists. I'm going to be brief because also Minister Mkhize needs to come in and tell us what does that mean from the side of his leading the fight through the health department. But just two things that I just want to say in concluding this and in thanking our scientists, that as the Department of Science and Innovation, we are really and truly committed to continue supporting and funding genomics, because the study of genes, genetic makeups and so on, it's a very, very important subject. For instance, globally, the region that has studied most its genetic variety is Europe. And the least studied region is the African continent. Yet scientific evidence points out that Africa possibly has got the single most concentration of genetic variations in the world. So it's clear that in South Africa, we are really in a pole position if we can put in more resources to do genomics, which will help us in future to deal with not just COVID-19, but other pandemics, as scientists are telling us that we've actually entered an age of pandemics, and that would be lucky if we finish the next 10 years without another major pandemic. So investment in genomics is something that is very important that we are committed to as the department. The second and last point I just want to make in addition, I think it would be proper that we really congratulate, whilst we congratulate everyone, all our scientists and their networks. But in this instance, we congratulate Sandile Cele, who is the PhD student and has been at the center of these new discoveries and so on. I'm raising this because he really must be an example and a role model to other younger people who have got aspirations to become science. And for me in particular, this is very important because I have been insisting in my department and with our entities, that scientific work must continue to be a training space so that we are able to produce especially more Black researchers and women researchers. So we really want to congratulate Sandile and say we are inspired by your work. I don't know, Ishmael, whether he is able to show his face if he has joined this, because I think this should act as an encouragement to other young people who are aspiring that it is indeed possible. I don't know if Sandile is here. It would have been good for him to show his face so that everybody can actually see him. He really is a very inspiration to our young people. Thanks, Ishmael.
Thank you very much, Minister. Sandile, are you here? Probably Minister will try to get him before we end our media briefing. May I take this moment to invite the Minister of Health, Dr. Zwelini Mkhize, to make his reflection. Over to you, Minister.
Thank you very much, Ishmael, and thanks to Minister Nzimande, Minister for Higher Education, Science and Innovation, the Directors Generals, and the eminent scientists at KRISP, and the entire consortium, Professor Thuledo Rivera, Professor Segal, Professor Mlisana, and all the scientists, members of the media and fellow South Africans. This is indeed good news for all of us. It's a kind of reflection of the good work that has been done by our scientists. This goes back to a collaboration between the BGI, which was known as Beijing Genomics Institute, and South African Medical Research Council, which culminated in the opening of the Medical Research Council Genomics Center. So this, by the time this center had opened, it had sequenced 6 genomes, which included a sequence of a child that was in complete HIV remission, and a sequence of a woman with highly resistant breast cancer. And at the time, scientists were extremely excited about tackling some of our most pressing health problems, such as HIV, TB, and communicable diseases like cancers, and so on. I want to commend and thank the Minister for Science and Technology, Higher Education, Science and Technology, and the department for supporting our research community. In particular, the work that has been done has been greatly supported by this department. We can say that as well, that it's very encouraging work, which is a work of a consortium. And we must also say, whilst all of them have done extremely outstanding work, we have also been inspired by the presence of a young budding scientist there, Sandile Cele, for the work he has done, and I hope that he will show his face. But at the same time, I think it's also important to say that South Africa has emerged as amongst the world leaders in surveillance genomics, which is something that's really strengthened our ability as the South African government to respond to COVID-19, which is led by science. You are aware that we have a very competent team of immunologists, virologists, and microbiologists, who are part of the ministerial advisory committees linked to a whole network of specialist clinicians, and that whole body of knowledge sits behind what decisions cabinet has had to take at a lot of in a lot of instances. And this is for us very, very important, because we have science basically leading, showing us the way. And so each time we move on the basis of the latest available scientific research findings. I think the other advantage South Africa has demonstrated amongst the world leaders in science and research has been the fact that we have got high level of competence in terms of scientific research, but combine that with the transparency with which we have handled the issue of COVID-19, it actually makes us to be able to go out there and share news even before many countries have been able to find this out. And we hope that that does not become any form of disadvantage, because South Africa, some of the countries tend to find out what's happening and they might feel that it's a problem that's emanating from South Africa, when in fact it may well be more than just South Africa, as we have seen on the map, showing more countries that are showing these variants. And so we see that now some of the countries may not even have discovered. In our case, KRISP was amongst the first to discover the variant ahead of the UK. And so we now have seen that the world is also looking at the research and the findings that are coming from South Africa as a way of looking at acknowledging the role and the leadership that South African scientists have been able to demonstrate. So we want to commend them for that. Because this is the team that first showed us that the change in the second wave was actually driven by the predominance of this variant 501v2. That was more transmissible. And then, of course, it ended up with a much higher level of hospitalization than before. They were the ones who were actually able to show us that as we're moving further and further away from the first wave, we were actually seeing the dominance of this. And as we have seen the demonstration now, that this variant 501v2 was actually almost responsible for all the 99% of the genetic sequence that was discovered on the various viruses that actually were causing the latest wave. This to us has been a very, very important and enlightening thing. But this is the team that was able to also demonstrate to us that the serum from the first variant, people that were convalescent from the first variant, was not effective in neutralizing the second variant. It did shock us, actually, to be honest, when we found that out, and we realized that now the cases for reinfection were now quite open, and we have seen many people have succumbed from a reinfection as a result of that. And this was discovered already. By the time it happened, we were already aware that this was going to be one of our challenges. They also indicated that the serum from those convalescents was not effective, which therefore meant that there was likely to be a challenge with some of the vaccines when it came to the second variant. Now, there has been a collaboration as well as they were doing the clinical trials with the genomics team to look at how these particular vaccines fared in relation to the South African variant. And so the latest trials that had to do with Johnson & Johnson, Novavax, and AstraZeneca was actually benefiting from this kind of knowledge, which for us has been a very, very helpful thing as such. Now, this discovery that the serum from the convalescent patients from the 501v2 neutralizes the virus, the 501v2 variant, as well as the earlier variant, is a huge boost into the knowledge, the scientific knowledge that is necessary to control COVID-19. And this actually opens up a basis for a conversation, which we are now already started having with the manufacturers of the various vaccines, because in those engagements, we're now actually saying the vaccines need to actually target the bigger problem, which is the 501v2. When we noticed that AstraZeneca was able to neutralize the variant which was in the UK and in Brazil, but not the one in South Africa. And therefore, it means therefore more work needs to be done in this regard. And everyone, all eyes are going to be on South Africa as to the level of success recorded in this regard. And I think it's an important issue for us to say, therefore, we are going to be engaging, and we hope that there'll be a lot more collaboration with our scientists with regard to this particular work. And so we are going to be approaching the vaccination program with this knowledge in mind. And we believe that the guide that you have seen here being tabled, which is showing clinical efficacy of vaccine, give us a guidance that we will have Johnson & Johnson, Pfizer, and Moderna, and so on in a kind of descending order. We expect that this work might show various changes as the research gets done, but at this point, we believe that South Africans will be getting the vaccines that are best suited for our situation. We have indicated that our situation was different. It's not that the issue of AstraZeneca was our problem, but because of the findings we had, it requires us to do a bit more work. So we believe that it's important for us to acknowledge the fact that our unique situations has required us to behave in a particular way and approach issues in a particular way. So this gives us what we call the focus on precision medicine, where it might be something that's effective overall, may also need particular tweaking to deal with the local circumstances. So we hope that this has been demonstrated is actually going to help us as we move on advancing in the area of surveillance genomics, as well as looking at how we utilize that and apply this science in the improvement of the vaccine, so that as we move towards the latter part of the year, we should be getting vaccines that are much more targeted to be able to deal with this variant, and the previous ones may be helpful in dealing with the variants for the future. And from that point of view, we should expect that our plans, the vaccination plans, will obviously be tweaked depending on what we are finding at this point. And then with those words, I want to once again congratulate all our scientists, the consortium on genomics, Professor Tulio de Oliveira, Professor Segal, and all the other, Professor Mlistan, and all the other leaders who are part of this, and also thank all our members of the Ministerial Advisory Committee for all the support that they've given, and thank the Minister and Department of Higher Education, Science and Technology for all the work that has been done. I think we're much, much better off with this information, and moving forward, we hope that South African scientists will continue to lead the way and help us guide not only South Africa, but the whole world in terms of how we can, as humanity, deal with the problem of this invisible enemy, COVID-19. Thank you very much.
Thank you very much, Minister Mkhize. We now come to the section on question and answers. I will ask my colleague Takalani Mukwebo to read the questions, and ministers and the panelists will assist in responding to those. We also welcome Professor Salim Abdul Karim. He can also participate in instances where certain questions will be posed to him. Prof, we don't know if there is any input that would like to make before we take questions. You are welcome to do so.
No, I've got nothing more to add. I think it's been all well said. Thank you.
Thank you very much, Prof. Over to you, Taki.
Thank you. Thank you very much, Ishmael. The questions that I have here, I have one question here from Christian Duplessis, or there are two questions from Christian Duplessis from Network24, and the questions are directed to Prof. Tulio. And the first question is, what is the chance that the virus will mutate again to the extent that antibodies will not be able to neutralize the potential new variant? And the second question from Christian Duplessis is, why is the immunity from vaccination better than immunity from natural infection? And the following question is from Tamar Khan from Business Day. And the questions are also directed to Prof. Tulio. And the first question from Tamar is, has the research we presented today been published either on preprint server or in a journal? If so, please provide the details. The second question, what do your results mean for individuals who recently had COVID-19 in South Africa? How confident can they be that they are protected from future infections? And we have question from Claire from Radio France. And the question is, accept the test on the 7 people in the study of PR more. Are there any more signs of the neutralization of 501YV2 on the other variants discovered in the world? And we have Sisona from News 24. And Sisona is asking 3 questions. And the questions are also directed to Prof. Tulio. And the first one is, what do the presented developments mean in the fight against the virus? And the second one, what are the key facts an ordinary person who is not knowledgeable about virus should know from the developments presented? And the last question from Sisona is, what do the results mean for people who tested COVID-19 positive? And Ishmael, if you may allow me maybe just to finish the 3 last questions so that they can be taken at the same time, because they are the last 3 questions that I have from my side. And one is from Sipamandla Goge from ENCA. And his question is, why has the South African government not dismissed the labeling of the 501YV2 as South African variant? And so far, it seems only Prof. Karim and Prof. Tulio today that have strongly rebuked the labeling of this as the South African variant. And we have a question from Sophie Mukwena from the SABC to Minister Nzimande. And the question is, how far is the Department of Science and Technology in studying a process or research in building capacity for development and manufacturing of vaccine that will help us deal with future pandemics? And the last question is from Elsabe Brett from Freywigbald. And the question is, what does this mean if you have not been infected with COVID-19? And what does it mean for the current vaccines? Will they need to be updated? That's all from my side for now, Ishmael. Thanks a lot.
Thank you very much, Taki. May I take this moment and ask Professor Tulio to lead us in response, then the ministers will come towards the end. This is the only session where we are going to take questions. We will not take any further questions, and would advise members of the media to send further questions to us. We'll give the details at the end of this briefing. Thank you very much. Prof.
All right, we're going to come out of that question and answer session now. Of course, the genomic scientists...
For the questions, yeah, I have taken note of the majority of them. Yeah, in case I miss one, please remind me. And I'm gonna be asking to some of my colleagues that present, and also my colleague Salim Abdulkarim to comment, because some of them will be beyond my knowledge. Yeah, but the first one, it is I can answer that. Yeah, what's the chance of development of more mutations and appearance of new variants? Yeah, so basically, yeah, this virus tend to evolve. That's what viruses do. Yeah, and that's why it's so important to have a very active genomic surveillance network. Yeah. What it means that the majority of the mutations that the virus develop, they are what you call silence mutations. They don't have an effect on changing the properties of the virus. Yeah, only when you have like drastic changes, similar what's happening in the variants. Yeah, accumulation of multiple mutations, 20, 30 mutations, and many of them in proteins that are associated either with transmission or neutralization, that's become a problem. Yeah, and that's why once this happens and these variants appear, they tend to dominate very fast the infections, and that happened both in South Africa, neighboring countries, but also with the one that was discovered in the UK, both in the UK, but also neighboring countries. And now we keep discovering new ones, both last week in Uganda, another one potentially in California, another one in New York. So we know that these mutations will happen and potentially variants can appear. And the main take-home message is that, yes, it is possible, and that's more than ever. It is important to try to control transmission, to don't let this virus basically outsmart us, and to increase vaccination as fast as possible. And I will steal a sentence from Professor Salim Abdul-Karim that he said, no one is safe until everyone is safe. Yeah. So to just summarize, it's possible to have more mutations and variants. But one thing that we'll do in South Africa, and that's what Professor Koleka Melisana highlights, is this very quick turnaround time of doing genomic surveillance. But the best would be to try to decrease transmission. Yeah. The next question, if it is okay, I will pass to Professor Salim Abdul-Karim, and it was a question associated like why the vaccines eluded much higher level of antibodies than natural infection, and I would like Professor Salim Abdul-Karim to please answer that for me.
Thank you very much, Professor De Oliveira. It's good to be here with you and wonderful to hear the research findings. I think, put very simply, we did not have, until the COVID-19 pandemic, the use of this new platform of technology using mRNA. In fact, the vaccines that are manufactured using mRNA technology and live viral vector technology have never been used effectively in vaccines before. So these are new technologies. And what we are learning is that the technology platforms that use mRNA, that's being used by Pfizer and Moderna in their vaccines, that when mRNA is injected, it elicits antibodies at a very high level. It elicits antibodies that are compatible with the highest levels achieved from natural infection. So that's a good sign, because it means that even if your antibody activity or neutralizing activity is compromised, as we see with many of these variants, if you start off with a very high level, it gives you greater protection. And in fact, many of the vaccines are producing antibodies at those kinds of high levels in COVID-19. So that's a good sign, and it bodes well, not just for the mRNA technology, but in fact for most of our vaccine technologies at the moment. Thank you.
Yeah, thank you, Professor Abdul-Karim. Ismail, should I continue with the question? Yeah, I will pass to some members. Yeah, so I just would like to ask potentially both Penny Moore and Alex Siegel, and maybe if Koleka want to comment there. Very simple. Yeah, what's the take-home message for people that already have been infected on this high neutralization?
Yeah, thanks, Julia. Alex, should I have a go first? So the take-home message is very much what take-home message has always been with people who are infected. We've known for a long time that people who are infected develop good antibody responses. But what we don't, there are many questions we don't know. We don't know how long those antibodies do last in people who've become infected. And we don't know how much antibody is enough to protect you from reinfection. So while we assume that people who've become infected with the new variant are protected to some extent from reinfection, we don't know how long that protection will last. And so we strongly encourage people who, even those who have been infected and no doubt have antibodies shortly after they've become infected, to still pursue the same routes of protection that we advise people who've not been infected. So non-pharmaceutical interventions, continue washing your hands, continue sanitizing, continue wearing masks, because there are many questions that we don't understand. We think from this data that people who have been infected with the new variant may be better protected against other variants and potentially the old variant should they be exposed to that, which is not likely in this country at the moment, as Tulia pointed out. But we think that those people may be better protected against other variants than they would have been had they been infected with the original variant. But as I say, we don't know how long that protection will last. So the bottom line is that you need to protect yourself in as many ways as you can and not assume that your antibodies, should you have been infected, are good enough to keep you safe again.
Yeah, thank you, Penimo. Yeah, I don't know if Alex Siegel would like to add to that. I can just add that, please, Alex.
Yeah, first maybe I'll explain Sandile's absence. So Sandile is doing science right now. So he's doing experiments in BSL-3. And I think for a virologist, I mean, this is the best thing you can do. So, you know, he's young, he's excited. And I think that's for him doing the experiments, because he has a lot more, there's a lot more coming from him. It's not his last contribution. So as far as what Penny said, I completely agree. I think the first rule of virology is don't underestimate your virus. And we have certainly underestimated this virus. And not we specifically only here, but around the world. People didn't realize it's going to evolve. So we think we understand it now. And we think we might have a solution, but let's not get complacent, right? We have to still maintain all the procedures that will prevent infections, and we have to be very careful and see what the virus throws at us. I think the news that's coming out here is good, but time will tell exactly how things will pan out and where the virus shifts. In fact, that's one of the things Sandile now is trying to predict, is what's the virus's next move. It's a chess game, right? Okay, we think we got a good move, but there might be a counter move. So not let our guard down now. Don't expect that because you had this variant that you are protected, because there might be surprises.
Okay, thank you. Yeah, so I would just continue. It's okay. Tell me when I should stop. Yeah, I think that for the next question, it's very appropriate again to Professor Salima Abdulkarim. Yeah, would be like what these new results mean for potentially developing of more effective vaccines or to the current vaccines. Does that change anything or not at the moment?
Yeah, thanks, Professor De Oliveira. So when we say that this new variant is able to generate immunity that is quite potent against itself and against other variants that we have been able to test it, that's fundamentally saying that if a vaccine is built on this new variant's strain capabilities, or what we refer to it as epitopes, in other words, the pieces of this variant that are immunogenic, if we make the next version of the vaccine based on that, we can expect that there's a good chance that vaccine will elicit good immune responses that will protect people from getting 501YV2 and probably several of the other variants. So that's the implications of this finding. This finding is basically telling us that we have a good prospect of success if we make a new vaccine based on it. Now, such a vaccine has already been made by Moderna. They've already done so. It's already now been sent across and it's going into human studies. Pfizer is doing similarly. AstraZeneca is doing similarly. Johnson & Johnson are doing similarly. So they are all using the 501YV2 as the variant that they want to try and target. They are using the parts of it that are immunogenic, and they're putting it into their new vaccines. So when those vaccines become available, they will be protective. Now, you have to understand that doing all of this takes time. So it's not something you can just do overnight. With the mRNA platforms, you can do so more quickly. It takes about 8 to 10 weeks, which is why Moderna is so far ahead. And now they will go into human studies to look at immunogenicity. For live virus vectors, those are AstraZeneca, Gamaleya, and Johnson & Johnson. Those will take longer. It takes somewhere in the region of around 3-4 months to make a new construct, because you're actually having to take a live virus, insert a new sequence into it, and then look for immunogenicity. So it takes a bit longer to get there. But eventually, by the end of this year, pretty much all of the vaccines, is my guess, will have the 501YV2. Not because they specifically worried about the virus coming from South Africa or from the other countries where this virus is present, but because a key mutation in the 501YV2 is actually also present in many other variants, like P1 from Brazil, like B1526 in New York, like Cal20 from California. So there are many of these different variants that contain key elements and key mutations that we have in 501YV2. And so if it works against 501YV2, there's a good prospect it would work against those as well. I'll stop there. Thank you.
Yeah. Thank you, Professor Salim Abdul-Karim. Yeah. And if it's okay, I'm gonna ask Professor Koleka Milasana to answer the next question. Yeah. So was a question like not only what these results, but what genomic surveillance is important to fight the virus in South Africa. And if you can highlight, Professor Milasana, why this genomics is important, not only to understand the vaccine, but other areas. Yeah.
Thank you, Tulio. I think actually just the results that we're presenting today beautifully demonstrate the importance of this work. Firstly, the fact that it is not just one particular, research unit within the country, but we've actually managed to do surveillance across the country. We therefore can monitor how the virus behaves within the country. And as a result, because of the collaboration and the network that we have actually shown, even with these results, we are then able to see what's happening across. And now we're getting results that show us what is happening within the variants that we have in the country, but we can actually test this even further with variants that are emerging in other countries. So really, I'm sure everybody can appreciate the importance of the network and how then it's going to be critical. I mean, people are asking, are we likely to see more mutations? And you've correctly said, yes, we're likely to see more. And that's why we need to continue with the surveillance, because we don't know what the next variant is going to look like and how it is going to behave. But what we have this far, and the great news that we're sharing today, is that the current variant that has been circulating in the second wave seems to have better protection compared to the first virus and even to other variants that have been tested. But what I also just want to remind everybody is that, as Professor Moore, illustrated, obviously this is limited information. So they've just tested it to the first virus as well as its own variant and just one or two other variants. So we don't have data for the other variants in other countries, but that is work that is ongoing. And so we'll be able to see how much more potent are these antibodies from the recent variant. Thank you.
Thank you very much, colleagues. In the interest of time, ministers are having to go for other engagement. I would like us probably, Prof. Tulio, if there's only one question left, we can respond to that. Then we give Minister Mkhize. And then before he comes in, let me use this opportunity to share the picture of Sandile as requested. This is him that we've been talking about. Thank you very much. I'll now hand over to you, Prof. Tulio, and then Minister Mkhize will come thereafter. Thank you.
Yeah, Ismail
Okay,
Sandile is on the line. He should be with us.
Yeah. Okay, so as we wait Sandile, I see Sandile every day, yeah, and he's a wonderful guy. I hope he can talk to us, yeah, and he's very handsome also. But as we come to him, to be honest, the next two questions were for our ministers, yeah. One of them, it is to our Minister of Health, that why South Africa has not been dismissed by calling the variant, yeah, and only Professor Karim and Tulio did that. First, is not true. I think that our minister was some of the most vocal in the beginning, but he will answer himself and his end comment. And then the next one, the question that in the line was for our minister Nzimande. Yeah, how far is the DSI in investing on not only the basic science on vaccines, but potentially evaluate and manufacture them? Yeah, thank you very much. And
thank you very much, Professor Tulio, and the panelists. I think it's been a very wonderful exchange, and thanks to the media for the questions. Yes, it is not really true that we have not protested or objected to the use of this name of the variant as a South African variant. We said from the beginning that what we need to do was, the fact that it was found in South Africa, first, it does not mean that there's something specific about South Africa and the variant. It just happened to be here, and that other countries are going to be picking up various variants, and therefore we also believe that it's a matter of time. Our countries where it might have pre-existed before South Africa but they may not have discovered it. So we don't agree that it should be labeled as though it's a South African creation. Yes, it was found in South Africa. In the same way, we don't call COVID-19 the Wuhan virus or the Wuhan variant. Nobody says so. And even though most of the variants that came here came from Europe and the US and other parts of the world, we never called it such. And so I think it's important for us to just keep making the point that South Africa cannot be stigmatized on the basis of a variant. And also, that's the point I was making when I said transparency and high technical expertise is the basis on which we have to be known to have actually found this virus. It is not because that should become a disadvantage to South Africa. It is our strength that we are transparent and that we communicate issues very quickly. We are also, we've got high scientific expertise that allows us to be able to get findings that are helpful in dealing with the problems that are facing the world. And we collaborate with various players in the whole world. And on that basis, South Africa cannot really be stigmatized on this. So we call this 501 V2. And every other variant has got a technical name. And so we don't have a South African variant. The variants are COVID-19 variants, but those variants are found in different parts of the world. The same one here, you have seen that it's actually already existing in 48 countries. And all of that means that we will continue to make it clear that South Africa can't be stigmatized on the basis of the variant. But it has helped us to be able to give guidance in terms of what can be done to improve the potency of the vaccines that are being manufactured. Thank you.
All right, the Health Minister there, Dr. Zwelini Mkhize, just giving some responses.
Mr. Nzimande.
Yes, thank you very much, Minister Mkhize. Thanks, Ishmael. And to to our scientists as well for the questions they've fielded. I just want to say to Sophie Mkwena, in answer to her question, that I'm pleased to say that we have already mapped the vaccine value chain, what it involves, the various steps of it, and we know how and where the competences are in our institutions and in the country generally, and where the gaps are. So we've done that initial surveying of our capacities and gaps. We are now in the process of developing a business case, which is aligned, obviously, in the case of COVID-19 to different vaccine platforms. So we can decide how we get strategic partners to fill those gaps. And of course, what would the required resources be? But in addition to that, the Interministerial Committee on Vaccines that is led by the Deputy President has already set up a work stream that is dealing specifically with this matter of developing South African vaccines, including a COVID-19 vaccine. And it's through this platform, of course, of the IMC that will be monitoring and reporting progress through to Cabinet. So this is already very important groundwork that has been laid for this. But also what I just need to point out is that one of my goals in this portfolio that I'm holding, and as part of the lessons from COVID-19, is to work towards building a center for the study of pandemics in South Africa. I've already started some informal engagements with Sefako Makgato University to begin to look into this possibility. But secondly, I wish to also point out that as from April this year, I will be launching officially the feasibility study for the building of the new University of Science and Innovation, as announced by the President, which will be built in Eguruland. I would also like that one of the early tasks that this university would be given is actually in this area of the study of pandemics. And what I am looking at is the setting up of a multidisciplinary center, because pandemics are a medical, a social, or even a political phenomenon. So it's very important, as an important lesson from COVID-19, that the Department of Science and Innovation, obviously working with my other Department of Higher Education and Training, we actually begin to build capacity for the study of pandemics in our country, given the future that we are faced with, and pandemics in their multidisciplinary nature. So that's the kind of work that we have done and that we are doing, and some of the immediate plans that we are having, so that as South Africa, we do believe that we actually do have capacity to do a lot more. And of course, working as part of the broader African continent and other developing countries, in partnership, of course, with scientists in the developed world who are willing to actually work with us to actually be able to do this work. Because without developing this capacity, which is why we are pleased the President has been insisting on this, we are going to face many problems, as a country and as a continent in particular. I hope that answers your question, Sophie. Thank you very much, Ishmael.
Thank you very much, ministers and the members of the panel, colleagues, members of the media. There are still some few questions that are not answered. I've requested the members of the media to send them to my email. We will respond to those questions. But this now brings us to the end of our media briefing. Thank you very much for all who participated. Ngiyabonga.
The Kingdom of Lesotho has adjusted the COVID-19 mitigation strategy down to purple.
Thank you very much.
Thanks very much, Kabazel.
Yeah, thank you very much, ministers.
Thanks very much. Thank you very much.
Thanks to the whole team. Thank you very much. Well done.
Thanks to the entire team.
Thank you.
Keep up the good work and keep on keeping safe, everybody.
Yes, thank you. Yeah, we hope next time that we don't come with more bad news. Let's hope the good news.
Yes.
On the sciences.
This is already good enough.
Yeah.
Thank you.
Thank you very much. Thank you very much. Thanks to Ismail. Cheers.